Hello students, I am Dr. Akshita Dhaliwal from Modi College, Patiala. Today I am discuss with you role of Agrobacterium tumefaciens in gene transfer method and specifically the function of their genes. The introductory part of this topic has already been discussed in previous video. So this is again the diagram of TI plasmid. The only tDNA region is transferred to the plant through the wound. The plant releases phenolics through the wound, which is recognized by the bacteria through the Vir-A proteins. And these proteins are present on the surface of bacteria. So in this diagram, you can see when the phenolics are released through the wound, they are recognized by the Vir-A proteins present on the surface of bacteria. These Vir-A proteins activated by the signals produced by the plants and binds with them, and it activates the Vir-G proteins downstream. And these Vir-G proteins act as a transcription factors and responsible for the transcription of Vir-G genes. After the activation of Vir-G genes, Vir-D1, Vir-D2 and other proteins are transcribed. The other types of proteins are explained in the form of chart. So this is the chart in which various types of Vir genes along with their functions are discussed. Like Vir-A gene, it is a kinase protein in bacterial membrane and act as a receptor for phenolic compounds released by wounded part. Vir-G activate other Vir genes and act as a transcription factors. Vir-D is responsible for virulence activity. So this is all types of Vir-G genes. You can read these functions later on, and these are easily can be identified from their functions. The list is continued. In this, when the Vir-D1 and Vir-D2 are formed, the Vir-D2 recognize right border sequence and Vir-D1 recognize the left border sequence and produces a cut, and Vir-D2 binds at 5' end. And the Vir-D2, which is present in tDNA, it binds with the Vir-E protein so that it can protect it from the degradation, and from these Vir-D2 transport to the plant DNA and attach to the genome, and it has gene of opine and cytokinin and auxin synthesis. Due to overproduction of auxin and cytokinin, it results in crown gall disease. So, the transport feature of this tDNA is used by the scientists in cloning experiment. So, they remove the auxin and cytokinin synthesis gene from the tDNA and inserted the restriction endonuclease site. Right and left border sequences remains the same. So as the restriction endonuclease site is present, we can add a gene of interest and can transfer this gene of interest to the plant cell through the same mechanism which I already told you in this video. Vir gene activates in the same manner as in normal cell. Thus, in this way, Agrobacterium tumefaciens is used in cloning experiments. I hope you can easily understand this topic with these videos. Thank you.